The title of the talk is the reading and interpreting genome activities, which sounds very big topic. But what I intentionally to talk about is the project, in-house project, which involved in the lots of genome-wide study. It is basically a project-oriented discussion. So talking about genome-wide study, we cannot ignore the genome biology. So let's start with a little bit background of genome biology. So far, it's been 60 years since DNA double helix had been discovered. Since then, we have sequenced so many genomes. This data is up to last year, 2013. We have sequenced 184, 83, 84 eukaryotic system. I want to highlight some model system here. The first one is the virus, which is about 10 kilonucleotides. The underneath is the yeast. Yeast is a widely used system which is being used for DNA replication and DNA damage and repairing study. Underneath yeast is a C. elegans worm. This is a star model system which has been widely used for developmental biology. And with this system, scientists had been discovered many basic biological processes, such as a cell programming death and RNAi, all those very basic biological system process had been discovered from this model system. This system actually produced at least so far 6 Nobel Prize winners, as I know. Next one is the fly. Fly is a system had been used for a neurobiology for the behavior study. And also, you see fish being used for cancer model, mouse, and of course, human beings, that's us. Here, one important message is, as the system become more and more complicated, you can see the genome has been increased, genome size has been increased. But there's one important message you should keep in mind. The number of genes doesn't seem to be an increase as the system complexity become more complicated. The more or less from a worm to the human being, it's roughly around 16,000 to 20,000 active genes, which makes the genome fully functional. So that makes people think, what is really going on with this, the system complexity? You know, when we know the number of genes doesn't increase, but the genome size really had a dramatic difference. That means the dark region of the genome really makes huge deal for the system complexity. So far, we know lots of different components of the genome. We can almost position every component we know in the genome. Not just that, where are they and how big are they? So where are we now? In terms of the characterization, positioning genome components, we have done majority of the work. The next big thing will be to study the relationship of the genome components. How all those genes, RNAs work together and how those things being controlled. So there are so many genes, non-coding RNAs, microRNAs work together to make a genome fully functional. In the real world, it's really hard to find something which is comparable to the genome in such a complexity. So what I have imagined, that the genome is probably just as a conductor or music master who can really put all those musicians and instruments work together to make a beautiful concert. The research project is probably similar to the chapter of the orchestra. So what conductor does is to read the music piece, to tell the musicians when and where you should come up. What genome does is probably similar. It's just to read those predefined script, tells genes, hey, it's time for your turn, such things. That's probably what the genome does. What we are doing as a bioinformatician is trying to translate those predefined script to the language which biologists can understand. So in this talk, I will mainly discuss the hypothesis-driven studies and the approaches we are using for the interpretation of the study. The first project I would like to discuss is that it's regarding the macrophage polarization. We know the macrophage has two different status. One is a silenced or naive macrophage. And after it being exposed to pathogen, it will become active, which is called polarization. In this process, there are many different things being involved in. What we are trying to understand is how macrophage being activated. During this process, what is working with this process? And first thing, of course, we would like to take a look gene expression profile. So this is a experimental design. We have the, this is the wild type naive state of the macrophage. This is after stimulus will become active polarized macrophage. And in this experiment, we also intentionally knocked down one important gene, we believe, which had been involved in this development process, and do semi-exactly stimulation to see what is going on. What we really like to know is the difference between the wild-typed one and the one with the one gene being modified. Yes, please.
So when you say important gene, it has a phenotype, which means you don't have an M1 polarized macrophage anymore, or?
That's what we would like to know.
Okay, but you
we know that gene had been involved in many developmental stages.
So nobody else.
But we don't really know if this gene really involved in the macrophage polarization. That's why we modify that gene.
Is it obvious from the phenotype, or you have to do like? Is it a
from experience from other studies?
Yes, and they claim they see a phenotype once they disrupt the gene.
Right. That gene had been reported has something to do with other things development. But we don't really know if this gene really involved in the macrophage polarization. Okay? Sorry, I couldn't release the name of the gene. That's one thing I couldn't do. Okay, this is a very typical factorization problem. We have here is 4 groups. We have Y-type group and a Y-type stimulated group. We have knockout group and a knockout stimulated group. We have 4 groups. And also we have, within this experiment, we have 2 independent factors, what I call independent factors. These 2 factors can live without depending on another one. You can do purely stimulation with Y-type macrophage, and you also can just do the knockout for those macrophage. You can do the experiment. It doesn't have to live together for these 2 factors. So here, the biological question is that, what are those crear-dependent genes? That's the question we are asking. And what are those stimulus-dependent genes? What are genes really just response to the stimulation? And what genes perform differently in between the stimulate? It depends on the crear. That's a hard term to understand. What do we call a term, interaction term? So it's really easy to understand what does the main term means. That's crear-affected genes, and also stimulus-affected genes. That's easy to understand. That's yellow line and the green line. But what really hard to understand is this red line, which we call the interaction term. What does that interaction effect mean? Which means whether the similar question you can ask when you do this problem, whether stimulus is equally effective for both crear and crear minus. Or, in other words, is there any difference in stimulus depending on crear? Any questions? So, did I? Okay. So I give a more explanation here. Use this gene as an example. Let's say in GeneX, we have 4 different groups. In one group, which is just the baseline, that's pure Y-type. Another group, it just be affected by factor A alone. And also, we have a group that's knockout group, which factor B alone. But for this group, which is actually the one gene, one expression value, which contains 2 different meanings. So if we want to know the stimulus-affected genes, we can just compare M1 to M0. If we want to know the factor B affected gene, we can just do this comparison. But for the interaction term, we really need to reference all other 3 conditions. That's why we need to determine how to tell the system how to get the expression value correct for the interaction term. So I said these 2 factors is an independent factor. However, in the reality, we also need to deal with some factors which really depending on each other. For example, here there is some type of factors which really embedded or involved in some dynamic process. This is a project we are still in the process. What we are doing here is that we are studying a very special biological process which called a Gamergate transition. You know, the ant as an animal, they usually form into a social group. A majority of ant species, if queen has been removed, the whole colony will be collapsed. It just fall into part. Everybody will die, workers will die. Each colony has one queen and many workers. Workers are doing many different jobs. But this end species we called a hypothesis saltus, which is a very special species. When queen dies or intentionally removed from colony, the workers will become very active. They will do fighting together and dueling, and all those type of behavior will come up. Eventually, they will get one winner. This winner will be the solo queen for this colony. So this supposed to be a worker now transit its role to a reproductive queen. This type of process is a perfect process for studying epigenetic controls, because the same exactly and switched rule in this process without change single code of the genetic information. So this has been used for this type of study. However, as you can see, ant has a pretty short lifespan for workers, which around 3 to 6 months. And the Gamergate transition has to take from beginning to the end, probably around 3 months. So these two types of process, aging and the transition process, really mingled. One will depend on other. And this type of factors, what we call the embedded factors or mixed factors. So when we studied this type of problem, we really need to think about how to set up a model to do the analysis, allow us to dig it out only genes which are with this transition, not with the aging, okay? So this is usually we do, when we do the problem data analysis, we first need to understand how are those factors work together. One is dependent on another one, or they are totally independent. And in clinical, there are many different things need to take care. In clinical situation, you really have many different factors being involved in. For example, when patient come in for some disease, you don't even know if this patient smoke a lot or drink and have some others being really involved in. So when we sort out all those things, now how to expand the genes has been involved in those interaction term. Here, I just give a simple example. In this case, when we take all those interaction genes, now we can see this group of genes really hasn't do much in the without the stimulus. In between the knockout and the wild type, it doesn't really change much. However, this group of the genes, after stimulus, it goes to total different way. This group of genes, we thought, might have lots of things to do with the polarization depending on that particular gene. That's a sort of interaction, the interpretation from this study. This is another project, a recent project we call the Fracking project. You know the Fracking story. It's an oil drilling story. So we know the fracking really a industry oil production method, but it hurts environment very much. Before, we really don't know, but from this case, we learn a lot. So what we did, we take the fracking water. The water had been taken from the spot where fracking drilling is doing. Take those water, put into the media to make the media, and then culture cells. After 24 hours, after a week, we harvest cells. And then take the, the harvest cells compared to the cells which cultured with just a pure normal media to see if there are gene expression changes. In this experiment, the first thing we have the time factor, which during this culturing time, no matter it's 24 hours and 1 week, the time as a culture factor, as a technical factor, it has something to do with the gene expression. If you have done the in vitro assay, you will know that. Even though you didn't do anything to the cultured cell, but to the end, after culturing, you compare the cultured cell without, just a settled down cell. There are so many differential expressed genes. So we set up that as a technical variation control by the baseline. Here, we also put into the dose factor with different dose of the fracking water. And the interaction term here, probably we can explain as an accumulation of the dose effect. So by this type of arrangement, to the end, we have been able to find out lots of differential genes. Water 1 is fracking water from Pennsylvania. This is somewhere from New York. And looks to me, well 1 is more toxic. It gets much more differential expressed genes. And also the number of genes are changed quite dramatically along this dose. This is a lower dose, this is a high dose. So this is interaction related genes. What I called is accumulated dose effect genes. Here, one thing you should know that when we do the gene selection, the positive genes really curved away when the number of reads are low. So we often get all these questions. Why can't you just do the fold cutoff for us to select those positive genes? So this is a very normal question from biologists because all they have been studied and told is a fold change cutoff. We know the gene expression data, especially for the sequencing reads data. The data distribution fall into what's called Poisson distribution. The Poisson distribution is really that when the reads are low, the uncertainty becomes much, much higher. You see, when reads are less than 6-fold, are in the low end, you really cannot tell the difference, even though they have 6-fold changes. So that's why we really cannot depend on the fold change for selecting the positive genes. This type of variation we call the model-dependent variation. It doesn't change no matter if you have technical controls or biological replicates, okay? So above, we discussed a couple of cases for gene expression profile. Any questions? Okay, now we are going to switch gear to discuss how gene expression being controlled. What is the gene regulation and the transcription regulation process? We know genome has two distinctive functions. One is replication, another is transcription. What we are going to talk about is transcription. How much so far we know about transcription? We have learned so far genome as a chromosome has two different status. One called a silenced status with a more condensed heterochromatin. Another is active status. We call it euchromatin. The silenced status of heterochromatin fall into 30 nanometer fibers. Active one fall into the 10 nanometer fibers. This is cartoon. However, this is also real. Here is the active status of the chromatin, silence status of chromatin under the electronic microscopy. What we call here is bits on the string. And this is a real image from the transcription. When I see this image, it's totally changed my thinking about gene expression. You see that I used to think about gene expression as a sort of a streamlined process. One RNA has been synchronized and then taken by another one. Here, what we are seeing is real parallel process. All those RNA sequencing fired up at the same time, almost at the same time. So you can see this Christmas tree type of image. And during the transcription, the first step is that the transcription initiator comes to the site, and recruit other transcription factors to form a transcription machinery. And it has been affected by either distal factors or proximal factors and get this transcription going. So to make a transcription process working seamlessly, a genome has to really follow two basic principles. One is precision, which being controlled by motifs. Okay, protein has to come to exact site. And find the exact location, start the transcription. Another principle is the efficiency. You really have to have a way to make that happen within a very short time. Think about that. If there is no efficiency, we will probably in a big trouble. After eating, we need insulin to digest things like glucose to make the energy for us. If there is no efficiency as a principle in the genome, then transcription may take hours to get to the exact point to start produce the insulin. So that's something really shouldn't happen. We know after eating about 30 minutes, we will have enough insulin to work for us. In the genome, there are also many different types of genes. One we called active genes, and inactive genes, and poised genes, 3 different groups of genes. Each of group of genes has been marked and arranged by those histone markers. For example, the poison genes, which really different from the inactive and active genes, which has been marked what we called a catenate 7 trimethyl and K4 monomethyl, which is really bivalent type of nucleosome within those regions. This shows how a poised group of genes being switched to the active state. So when a developmental signal comes into the site, it will find these bivalent nucleosomes and kick out a nucleosome, get the sequence exactly attached, then start the transcription initiation. This is a case we have been studied for more than 3 years, which we started a protein called ERK. This protein is along the MAP kinase pathway. When insulin or growth factor come to the cell, it will basically do the phosphorylation along this pathway, eventually activate the ERK and put the phosphorylation into this protein, make the ERK to be active. This process really have been studied quite, I would say, a very popular process has been studied so far, because this really related to how the stem cell be maintained in the self-renew status or switched to developmental status to become a different type of cells. So what we had found, this ERK protein, the phosphorylated protein, has sort of work to do with another 2 proteins, one called Jarid2, another called EZH2. Okay. Within the knockout status, you can see phosphorylated ERK become much weaker, and unphosphorylated doesn't change for both Jarid2 and ERK2. The question is, we know very well about Jarid2 and EZH2. These 2 proteins are the core protein of polycomb complex, which really maintains the level of those catenate 7 trimethyl. It has a catalytic activity to make the catenate 7 dimethyl to the catenate 7 trimethyl. Catenate 7 trimethyl is a key histone marker with those poised genes. Okay, this is a sort of information. Then the question is that, what does ERK do during the developmental process, and why ERK has been coexist with these 2 core proteins? Yes?
Sorry, were you saying that, what is the effect, the effect exactly of the polycomb to the K27 trimethylation? Like, is it activating another enzyme that then trimethylase?
I'm not quite sure, but what I have learned is the polycomb complex somehow can recruit a protein which has which can really catalyze the dimethyl to the trimethyl.
Okay, but no, but how does that happen? Because because ribosomes are generally not at the place where transcription is occurring. So how does that?
That's a great question, but really.
I mean, there's like an entire membrane.
I'm
I
sorry.
mean, it's in a completely different cellular compartment. So, like, is
The polycomb?
Yeah, as far as I can tell.
Polycomb complex 2?
No. Oh, no, I mean the
PCR2.
I thought you said polysome. I think I might be
Polycomb is not
Yeah.
a different compartment.
It's a polycomb replace. Yeah, it's called a polycomb replace complex. It's short as a PCR2 complex. Yeah, which maintains histone productivity. Okay, so then the first thing we would like to think about, if ERK has anything to do with a histone H3K27 trimethyl. Okay, we know the H3 trimethyl are along with the low expressors. Okay, in the gene expression profile, we have learned there are some of the genes with many number of reads, some of genes with very little the reads. Those are low expressors. The K27 really are occupied in those low expressors. Okay.
So when you're saying that binding, you mean actual binding of the protein? So is it like a Chipseq experiment or something? Correct?
For the histone?
For the histone, yeah, but what about there? Because you say binding.
ERK? Yes. Chipseq for ERK.
Chipseq for ERK.
Yeah. We did basically, this is well known. Everybody knows. H3K27 occupied the region which are really low expressed gene regions. But what we didn't know.
Related form of ERK or like?
I'm sorry?
Is it the phosphorylated form? Is it just ERK? What is this?
No, this is a K27 trimethyl profile. What we didn't know is ERK. You will see. So what we did exactly a Chipseq for ERK. And we look at same group of genes. Here, this is a profile of ERK. After the Chipseq, we see ERK occupied same exactly group of genes. This tells us ERK and K27, they may coexist. But we really don't know if they are functionally related, which we don't know. Yeah?
Opposite, right? And you mean it looks, the two profiles, if you compare the K27 profile with the ERK profile around the TSS, is it the TSS there? The zero?
This
They
is
do kind of the opposite, like the ERK is binding where you don't have that much as the K27.
Right. This is ERK peak. This is not TSS. This is TSS. So this means ERK are somewhere here. The peak region of the ERK are somewhere here, not at exactly TSS.
Okay, so then it would be, yeah.
Okay.
If the zero was the same between the two, it would be more obvious. Like it would be easier to compare.
You mean the how
The zero, yeah. If
the
it's like
distance from the ERK peak to the TSS? That's
like
your
if it's
question.
like the TSS of the gene, like the average profile that you have around the TSS of these genes, right?
So, okay.
both for K27 and ERK, in order to be able to compare the
Of
two.
course, yes. But people understand the K27 trimethyl has been really occupied the low expressed genes. Here, also tells us ERK are occupied those low expressed genes. Do you agree? Yeah, this is a message we would like to tell. Just give an impression. The ERK and the K27, they really coexist in the genome.
And the highly expressed ones?
Highly expressed ones is down here. Somewhere is red, you see? You don't have ERK, you don't have K27, you don't have ERK. That's the message. So, because from that study, we don't really know. We know they are sit together, okay? But we don't really know if they are functionally related. Then what we did is knock down the ERK to see what happens. So after knockdown, you can see that the K27 will go down. Is this K27 or this one? I couldn't read these letters. And also Jarid2 will go down. In the wild type, they are occupied. Okay, that tells us there's some functional correlation there. And the genome-wide, you can see that a K27 in this has been distributed like this way. And in the mutant, the K27 semi-exactly locus has been faded. And the same thing for the Jarid2. And this is the RNA, what's called POL2, polymerase, RNA polymerase 2, as a contrast. And then we also did a rescue experiment, okay? After knockdown, we rescue, we can see K27 level really recovered. This really a good experiment to tell us something ERK and K27 are functionally related. But we don't know how do they work each other, in what format, or does ERK really catalyze the K27 trimethyl? We don't really know. So we had proposed this model for the ERK function. If you are really interested, I would suggest you to read more these articles. But because I don't really have much time to discuss all these things, so please, if you could take a look at the paper, and if you have questions, please ask. So in this type of study, most of the time people will do the Chipseq. This is a regular Chipseq pipeline. We are doing these things almost day-to-day work for the Genome Technology Center. To the end, for the regular Chipseq, we will end up with a fragment resolution type of peak. This peak really covers the fragment we got from experiment. But we really don't know what is exactly binding nucleotides. Okay, the resolution is so different. We know the transcription factor has this type of scenarios. One is really narrow bind. Okay, just one transcription factor, find the DNA region and bind there. This is very narrow. And also, it could be find a widespread locus for binding. And also could have a dynamic switch. All this has been really seen in the past. You see, this type of binding is really sharp, narrow binding. These are dynamically switched and also widely spread bindings. These things does exist. So in our experiment, we would like to really to know what nucleotides have been bound by a studied protein. And this is a comparison between the two experiment protocol. One called regular Chipseq, another is ChipXO. What ChipXO can do is that we do exactly the same experimental operation, but ChipXO will add one more step. To use this exonuclease digest a DNA fragment from 5' to the end. When the enzyme digestion reaches edge of the protein binding, it will stop. It couldn't go on. So what left is all the reads. You can see from 5', it is exactly binding site of your right arm, if this is antisense. In the sense, you get left arm right on the edge of the binding region. Okay? So this is a protocol has been used for many of the ChipXO experiment. Now you can find out from web. I think Illumina has this. Here is something I would like to share is that to the end, you can see the reads. It's very precisely mapped to the binding area. Okay. So this is a publication from last year. Compared to the peak before people are dead. Now you can see very precise binding region. You can find out it's about 6 to 20 nucleotides. You can find exactly nucleotides. This is what we did. It's still in the work in progress. This is something we can tell, we can call, find out what exactly nucleotides we would like to look at. So second scenario is on the system level. How does this transcription factor binds to? The transcription factor tend to have different combinations work together to make different cells. This is from Robert Chen's presentation. Okay. So the different transcription combination will produce the different type of cells. The proving the transcription control in different type of cells is really next wave of biological studies. It's sort of a cutting edge. The frontier that lots of people works in this field are trying to approach. So what I am trying to, and a group of people are trying to do is that we are collectively produce the data and collect and organize all the data, and eventually would like to find out, first build these data collections and find out what are those combinations which can really make the stem cell developed into, let's say, muscle cell, liver cell, all different type of cells. This is something we are doing. So to the end, what I can tell is that genome and music are totally different. Everybody knows. But what I can say is that they really share some common. One biggest thing, I believe, they diversify word with only a few nodes. So then with this, I will stop my talk and take all the questions. I would like to thank all the people who worked with me in this talk and also routinely in the real life. Thank you.